At this point, the innate immune response has done an excellent job of either keeping our pathogen out in the first place, or if the pathogen got in, recognizing it as dangerous and producing cytokines that would increase vascular permeability and bring in more phagocytic cells, like these neutrophils here, which could then come in and help engulf the pathogen. Additionally, it's probably induced complement at this point, which will further allow for opsonization of phagocytes, remember C3b, as well as increase use of acute phase proteins and create your systemic non-specific innate response. Well, that's all well and good, but now it's time to engage the adaptive immune response. Remember that the adaptive immune response differs from the innate immune response in three main ways. First off, it's not immediate. It takes days to weeks to act. Second, it's highly specific. Remember, our innate immune response uses PAMPs and PRRs to recognize things that pathogens have in common. The adaptive immune response recognizes specific antigens. These are the portions of bacterial or viral or basically pathogenic microbes that our cells of the adaptive immune response are actually going to see. And lastly, because it's so specific, it has long-term memory, which means that the second time you see this pathogen, you'll respond faster and better. The adaptive immune response occurs through the expansion and differentiation of T and B cells. What do I mean by differentiation? Well, differentiation is the T and B cells' method of maturing or growing up. And just like as we grow up, we gain new skills and techniques, T cells and B cells grow up to basically acquire their superpowers that enable them to combat pathogens. So if you're a CD4 positive T cell, you're probably going to differentiate so that you're able to release cytokines, which will inform the type of immune response that we'll make against any pathogen. If you're a CD8 positive T cell, on the other hand, you'll become a cytotoxic T cell, which does literally what it sounds like. It basically kills cells that are intracellularly infected with pathogens. B cells, on the other hand, have a slightly different differentiation. They differentiate into a new cell that we haven't really talked much about yet, called the plasma cell. Plasma cells are the antibody-secreting form of the B cell and the fully differentiated form of the B cell. Another term for the term antibody is immunoglobulin. You may hear these words used interchangeably, so keep that in mind when listening to videos. Now, I've mentioned a lot of times that the adaptive immune response is antigen specific or highly pathogen specific, but what does that exactly mean? Well, remember that antigens are just the portion of the microbe that our T and B cells over here are actually going to see. And they can detect a much wider range of antigens than our innate immune response can. Remember, our innate immune response really only recognizes things that are shared across the pathogens that we might see. B cells can recognize proteins, lipids, or carbohydrates. T cells, on the other hand, can recognize only proteins, so really only peptides. And these are the macromolecules that the antigens are derived from. So your T cells are peptide antigens, whereas your B cells could be peptides or lipid-derived or carbohydrate-derived. Each T or B cell expresses many copies of the exact same receptor, either the T cell receptor, this guy here, or the B cell receptor. Note that the B cell receptor looks almost identical to the antibodies that I was talking about earlier. That's because B cells, or should I say plasma cells, can actually secrete their receptor, their B cell receptor, and that's what actually forms antibodies. T cells are not able to secrete their receptor, and it will always be surface bound. Each receptor is specific for a different antigen. So, for example, this T cell is specific for orange antigen, while this B cell is specific for green antigen, and so on and so forth, okay? The pink antigen over here will not be able to stimulate the orange antigen-specific T cell over here, and vice versa. Therefore, we can call this T cell the orange antigen clone, or this B cell the green antigen clone. So each T cell, because it expresses a T cell receptor, or many T cell receptors, I should say, that are all specific for orange antigen, is a clone. The same can be said for B cells. They might express many B cell receptors, but they're all specific for the exact same antigen, and therefore it is a B cell clone. When a clone sees its antigen of interest in the right situation, in the right context, it will massively expand. This process is known as clonal expansion, because while these T and B cells that saw their antigen will expand, these other ones that were maybe specific for blue or purple or whatever color antigen won't expand, because they do not have the same antigen specificity as the clones that are expanding. Expansion radically increases the number of cells able to target the antigen encountered. Adaptive immunity functions through two branches mediated by two different types of cells. Humoral immunity largely defends against extracellular pathogens and is mediated by B cells, and cell-mediated immunity, which largely defends against intracellular pathogens and is mediated by T cells. This process is called humoral immunity because antibodies can be transferred in the serum or humor from one person to another without any cells to confer protection. We saw this at the end of the Ebola epidemic when Dr. Kent Brantley, an American doctor, contracted the Ebola virus while working in Liberia. Luckily, he survived, and part of the way he survived was that he got a lot of really great treatment. But the other thing is he mounted a great immune response, and in doing so, he created a ton of antibodies. Well, much later, a traveler came to the U.S. and landed in Texas, and unfortunately, a nurse there happened to contract the Ebola virus from that patient. Well, Dr. Brantley was able to give some of his antibodies to that nurse, and she survived. This process is called passive immunity. Passive immunity is when a naive person, like our nurse over here, is a person who has not seen the pathogen before, is given antibodies from a person who had previously seen the pathogen. These antibodies protect them and help them to quickly gain the ability to combat the infection faster than they would have been able to do so on their own. Remember that B cells and antibodies can recognize many different types of molecules, including proteins, carbs, nucleic acids, and lipids. Antibodies are able to neutralize and limit microbes and microbial toxins present outside of cells, extracellular pathogens. And for this reason, they're really important at mucosal surfaces. Antibodies actually stop microbes found at mucosal sites and in the blood from accessing and colonizing our host cells and connective tissues to prevent the establishment of infection. Cell-mediated immunity largely defends against intracellular pathogens, and it is mediated by T cells. Specifically, it is mediated by two types of T cells, the CD4 positive T helper cells and the CD8 positive cytotoxic T cells. These cells work to either directly kill pathogens in the vein of your CD8 positive cytotoxic T cell, or to produce cytokines and other soluble molecules that will help other cells that are harboring the pathogen kill the pathogen, like your phagocytes. The standard nomenclature for referring to T cells and many other cells of the immune system is CD with a number, i.e. CD4, CD8. CD means cluster of differentiation, just so that you know what I'm talking about. Cell-mediated immunity, unlike humoral immunity, cannot be transferred in the absence of cells, and therefore is termed cell-mediated. T cells only recognize protein-derived antigens, or peptides. CD4 positive T cells can recognize fairly large peptides at about 14 to 20 amino acids, while CD8 T cells recognize much smaller peptides. T cells require a process known as antigen presentation. This literally means they want their antigens served to them on a silver platter, except the platter isn't silver. It's a protein called MHC, or major histocompatibility complex. And it's on the surface of either specialized antigen-presenting cells, in the case of CD4 positive T cells, or CD8 positive T cells actually use an MHC molecule that is present on all nucleated cells within your body. So that means CD4 positive T cells can really only communicate with antigens that are presented from specialized professional antigen presenting cells, whereas CD8 positive T cells could kill any cell in your body. Both humoral and cellular immunity can be acquired through active immunity, which is how most of us make and keep our immune responses. This is when an individual is exposed to antigens of a microbe, generates an active immune response, and clears the infection, and develops protection to later infection by that microbe through the establishment of memory. And that is the process with which we're going through this entire course for.